This is the Naked Genetics podcast, taking a look inside your genes.
Every day we get older, and whether you're desperately resisting the march of time or embracing the ageing process, most of us would agree we want to live as long, healthy lives as possible. We'll be finding out how genetics research can help.
You wouldn't want to live forever if forever meant living in misery. You want to be able to enjoy the things that you normally enjoy to the extent you do now.
Plus, making fingers with Alan Turing, growing lizard tails, and a long-lived gene of the month. This is the Naked Genetics podcast for September 2014 with me, Dr. Kat Arney, brought to you in association with the Genetics Society, online at genetics.org.uk. It's a sad fact of life that we all get older. And it's not just wrinkles and grey hair. There are many diseases of old age creeping up on us, from dementia and arthritis to cancer and heart disease. Cheery stuff. At the UCL Institute of Healthy Aging, researchers are investigating the genetics of ageing and how understanding the fundamental biological changes that happen as we age could help us live longer, healthier lives. I spoke to David Gems, Assistant Director of the Institute and Professor of Biogerontology, and started by asking him what we mean by ageing.
The word ageing is a very slippery term that causes a lot of confusion because it means multiple things. I mean, ageing can mean just that you gain more years, just in that sense, everything ages, everything grows older in the calendar
older,
time, Ageing can also mean, in a broad sense, any kind of change that happens over time. So, I mean, here's my coffee, and you could say that as my coffee gets cold over time, that's an age change, And so, in people, age changes can include all sorts of things. I mean, that can include maturational changes in temperament, and all sorts of things. But the ageing that I'm interested in is the deteriorative aspect of ageing, which biologists sometimes call senescence, to try to be clear about it. So the interest here is really in the biology of senescence. Senescence is something which is really not understood in terms of its biology.
This is the kind of the gradual conking out of our cells.
People working on aging, on the biology of aging, still argue about how to even define aging. There's different ways of defining it. I mean, I would say the thing which is really characteristic about aging, that is senescence, is the development of pathology, and pathologies that come from within. They're not caused by external things like germs or something. They're actually coming from within your own body. So there's some sort of process which gives rise to a load of pathologies. Those pathologies are, in the developed world, really the main root cause of death.
So these are things like dementia, heart disease, all these kind of problems.
Yeah, I mean, numerically, the overwhelming cause of disease in the developed world is aging itself. And I mean, there are a lot of aspects of aging people don't really think of as aging, but they are. So, for example, most cancer is actually develops as part of the aging process. And, dementia, as you mentioned, a lot of type 2 diabetes tends to get worse with age. Yeah, dementias like Alzheimer's disease, Parkinson's and others, I mean, it's bewildering how many pathologies there are. It's terrible, really, really.
And depressing.
Yeah, right. I mean, that I guess that's the human condition.
So what do we know in terms of, say, human bodies or other animal bodies? What's going on at a cellular or a genetic level as cells or organisms age?
So, one of the, as a biogerontologist working on the biology of aging, I mean, I can say with some authority that, we really don't know. And it's actually one of the things which is very exciting about working on biology of aging. It's the main cause of disease in the world, and yet
no one knows anything.
It's actually not known. Well, there's lots of ideas, and the state of play at the moment is there's a lot of research at the moment on the fundamentals of aging, but there isn't really an agreement about what sort of process aging is. So I think traditionally the most of the ideas about aging over the last 50 years have been based around the idea of aging as a kind of a wear and tear type process. So it's a bit like machines that wear out.
Your parts get a bit rusty, everything stops working quite as well.
Yeah, rusty is exactly puts a finger on it, because rust is metal oxidation, and one of the ideas is that we're living in this high oxygen atmosphere, and the oxygen burns it. So you have a sort of a process of oxidation in living systems, just like you do in mechanical systems. But I think in the last 5 years or so, this idea, there are many different theories kind of based around damage of one kind or another causing aging and how it happens, including telomere shortening, chromosome shortening is a kind of a damage theory.
Your chromosomes just getting shorter and shorter and shorter, and then they can't do anything else.
That's right, then the cells kick out of the cell cycle. I think at the moment, I really think there's been a kind of a watershed for this sort of aging damage paradigm, where people are starting to wonder whether it's really true, and there are some alternative theories that are appearing. So it's a very exciting period scientifically in terms of paradigm shifts and so on. I think.
So if you say there's lots of different theories, so what one is your favourite one, perhaps? And what are you trying to do in your lab to understand this process a bit more?
So the main approach that my lab and quite a few others have been taking to try to figure out aging is to look at the genes that control aging. So you don't really need to do any science to know that aging is controlled by genes, because different animal species have different lifespans. That is, their maximum lifespan. So if you take, for example, humans and our closest relative, the common chimp, our maximum lifespan is about double that of the common chimp. So we've evolved recently a much longer lifespan. It just shows how genes control aging. So what a lot of scientists are doing is using very short-lived animals like fruit flies, like these nematode worms, which is what I work on. They're called, the proper name is Caenorhabditis elegans. And they're great to study because their lifespans are very short. So C. elegans only lives about 2 or 3 weeks. So you can look at the genetics of aging in C. elegans. You can look for mutants, for strains of worm where they've changed genetic changes, which have different rates of aging, either that age more quickly or age more slowly. They're in a way the more interesting ones because they live longer.
So I guess in a worm, if you're getting one to live maybe a week longer, that's a really significant increase on a worm's lifespan.
Well, yes, it is. For example, there's a group in Arkansas who are currently the record holders for sort of extending lifespan in worms.
I love it's a competition.
Yeah, no, it's true. Because I feel strongly about this because I had the longest lived worm strain at one point. It was a 7-fold increase in lifespan, and these guys in Arkansas, they got 10-fold increase now in lifespan. But it's only a couple of months of life, extra life, and if you translate that to a human being, it's hard to know what that means. That could mean
700 years.
Is it 1,000 years, or is it an extra 4 months? It's actually hard to know how to translate it. The bottom line, though, is that what this tells you is that aging is actually plastic, and that's so important. I think of almost all the discoveries that have come out of this lifespan genetics, that basic observation that aging is plastic. It's not fixed. It is something that is alterable, is very, very profound, and has shaped a lot of thinking in the field and ideas about what the field can do. So, in terms of what we learn from that, what we hope was that we find a gene that controls aging, and then we find out what that gene actually does, what protein it makes, and what's the kind of biochemical cellular process that gene is affecting, and that should lead us to aging. What aging is.
How long would you like to live for?
Oh, I don't know. I do find that one thing about working in this field is that I tend to always want to not think about the fantasy aspects, but think about the realities. But if I had a choice, I guess I would like to be able to live long enough so that I could choose when to end my own life. And I don't know how long I would want to live. I think. Probably a lot would depend on how capable I was of reinventing myself and changing. It's unanswerable. Who knows?
That was Professor David Gems from UCL. Most of us are born with 5 fingers on each hand, but have you ever wondered how they get there? The answer was first put forward more than 60 years ago by mathematician and wartime codebreaker Alan Turing, who published a paper suggesting how molecules can interact to create stripy patterns, including the 5 stripes of bones that make up our fingers and toes. It's recently become clear that this kind of system is at work in developing limbs, but the exact identities of the biological players involved wasn't known. Now a team of researchers from the Centre for Genomic Regulation in Barcelona think they found the answer. I asked James Sharp, who led the work, about how he tracked down these mysterious molecules.
So the main approach was to distinguish between the cells that are going to form your fingers and the cells that are forming the tissue in between your fingers. During early embryogenesis, when your hand is forming, the hand is in fact a continuous plate of tissue. And some cells will become fingers, and the cells in between these regions will become the gaps between your fingers, although at these early stages they're still cells. So it's just a decision between 2 cell fates, making fingers or making the gaps. And the goal was simply to find which molecules show any sign of having a on-off pattern between these 2 cell fates as early as possible.
So you're basically looking for things that are set up in stripes at this very, very early stage.
Yes, exactly. And they could be genes or proteins that are either expressed or active either in the cells that will become digits or ones that were only active in the cells that will become the gaps. It could be one way or the other, it doesn't really matter.
So you track down these molecules. How do you know that they are the ones that are responsible for actually making this pattern and telling the fingers to grow there?
That's a very good question. The critical thing to do is to somehow make some predictions about what would happen if we manipulated these molecules to the pattern. So in addition to looking for the molecules, we also built a computer model, a computer simulation of the process based on as much evidence and data as we could get. With the computer model, we then made predictions about what would happen if you slightly repressed one pathway or the other, what would actually change in the pattern of the fingers. And then finally we were able to actually test that by taking little pieces of tissue, very small pieces of tissue, like less than 1 millimeter from early mouse embryos, and culture them in a petri dish, and actually apply drugs to reduce one pathway or the other, or both pathways together. And the very satisfying result was to see that when we did these experiments, they actually recapitulated the computer predictions very closely.
And obviously it's a really nice thing to know that this is probably what's going on, to be able to understand a biological system like that. But is there any way we can take it forward? I mean, how can this knowledge be used? Is there any aspect of, say, human health or tissue engineering that could be useful for this?
In the long term, understanding how tissues build themselves is the only way that we will be able to do proper tissue engineering. And what I mean by that is growing tissues in a dish, getting tissues to organize and build themselves so that they are functional tissues or pieces of organs or even whole organs. To be able to do that, we have to understand how the body normally does that. And that's essentially what this work contributes to. And unless we understand all parts of the story, all the basic principles by which tissues are organized, we will not achieve what must be naturally the long-term goal of this work, which is to rationally, consciously design tissues in a dish for various kinds of regenerative medicine. So I believe that this is inevitably going in that direction, but it's also clear that it's going to be a long way in the future.
One of the names that you've mentioned is Alan Turing. Most people think of him as a computer scientist, a mathematician. It's interesting to see that he's actually made such a contribution to understanding of biology, even from so far in the past.
He was really interested in how complicated and clever machines, let's say, arise in nature in general. And this led to his interest in the brain and in artificial intelligence and how you can calculate things, how you can work things out, but also to how these amazing machines, such as the brain or such as a body in general, comes into existence in nature. So I think that from his interests, these two things were not so different.
Professor James Sharp from the Centre for Genomic Regulation in Barcelona, and you can read my feature on Turing patterns in biology for free in the online magazine Mosaic at mosaicscience.com. And now it's time for a roundup of the rest of this month's genetics news. Writing in the journal PLOS ONE, researchers at Arizona State University have analysed the patterns of gene activity in regrowing lizard tails, which could pave the way for better regenerative technology in human tissues in the future. The scientists were studying the green anole lizard, which can escape from predators simply by losing its tail, then growing a new one over the next 2 months. But while lizards share many genes with humans, we don't share their incredible regenerative abilities. The team discovered that the lizards turn on at least 326 genes in specific regions of the regenerating tail. Some of these are genes involved in embryonic development and wound healing, as might be expected, as well as a biological signalling system called the Wnt pathway, which is also used by other animals that can regenerate their tails, such as salamanders and some fish. The researchers also identified a special type of cell that's important for the lizard's tail regeneration, known as satellite cells, which are also found in humans. The hope is that understanding the genetic recipe at work in the lizard's tail might lead to new ways to regrow human cartilage, muscle, or even spinal cord in the future. The day when we can grow fully functioning organs in a dish may have come a step closer, as researchers at the University of Edinburgh have managed to create a mouse thymus in the lab. This is the organ responsible for making T cells in the immune system, and transplant it into an animal. Publishing their findings in the journal Nature Cell Biology, the researchers took mouse embryonic cells called fibroblasts and used a gene called FOXN1 to turn them into thymus cells. When these were mixed with other types of thymus cells and transplanted back into a mouse, the cells organised themselves into a replacement organ that functioned the same as a regular adult thymus. It's the first time that scientists have made an entire living organ from cells that were created outside of the body by reprogramming. Although the work's currently still at an early stage, the scientists hope that their lab-grown cells could one day form the basis of thymus replacement therapies, or even lead to personalised T cells grown in the lab to help people with faulty immune responses. If you want to find out more about those stories, the references are all on our website. That's nakedscientist.com/genetics. You're listening to the Naked Genetics podcast with me, Dr. Kat Arney. Still to come, we'll be finding out what progress has been made in treating neurofibromatosis and unearthing a long-lived gene of the month. But first, we return to our age-old topic at the UCL Institute of Healthy Aging. Student Michael Shannock has been studying fruit flies carrying an altered version of a gene called glycogen synthase kinase 3, or GSK3 for short, which have longer, healthier lifespans than their normal counterparts. He's been trying to find out why.
So the flies in this lab live normally the best part of 2 to 3 months. On average, the flies that we were working on with this manipulation of this protein were living about 10-15% longer than the average fly.
So if you translated that to a human, that would be sort of living an extra, I don't know, like 10 years, I guess?
Yeah, if you took a direct correlation to humans, it would be about 10 years. But of course, a fly is not human. But there's some other promising strategies as well, which, for example, you've got mice, and we've known this for nearly 100 years now. Mice, if you give them about 60% of what they normally eat, they somehow live 30% longer. So that's quite nice. Obviously, we don't want humans to go starving themselves. And there are some people who practice this. It's called calorie restriction. It would be nice to have a pill that does the same thing without having to actually physically starve, cut out things from your diet.
Because food is nice.
Absolutely, we love to eat,
Food
right?
makes me happy.
Exactly, we love to eat. We love to eat things that are normally not very good for us as well. So it would be interesting to see if we could find a way of mimicking this restriction of calories in a way that doesn't actually affect the quality of life.
What exactly is the protein, the gene that you're looking at that's faulty in these flies?
So this protein is called GSK3. It's quite central. It's got a diverse range of things. So it controls survival of cells. So it actually promotes the survival of cells, which is great in one sense.
You
but also
do want
don't
your cells to live longer if you're going to live longer.
Exactly. So if your cells are healthy and living longer, then obviously your organs will live longer and you will live longer. So that's the idea. Obviously, there's a danger that you don't want to have cells living so long that they're actually replicating uncontrollably, because then you get into territories of cancer, which is not good. On the other hand, this same protein, GSK3, also controls cell death. So it can actually promote what they call the programmed cell death. So our body has ways of shutting off cells that aren't working very well. And actually the interesting finding, the main finding of my study, was that this is both specific to different areas of the fly. So having more of this protein in certain areas of the fly is worse off, and having it in another area of the fly is better off. So it's very tissue specific.
What have you discovered about it that makes the flies live longer?
So one of the things we were interested in, and this is something that's kind of common in aging research, is to try and stress test the things that you're working on. So you see, how do they react to different stresses? It might be in terms of starvation, how do they react to starvation, so no nutrient at all. And actually these flies were kind of sensitive to starvation, which was interesting. Then we also gave them a different stress in terms of what they call oxidative stress. So oxidative stress is basically in the process of breathing and respiration, producing energy from food, basically. You have a byproduct, this thing called reactive oxygen species. These are quite nasty chemicals that go out and damage DNA and just generally not very good. We have systems in the body that actually mop these up and kind of prevent the damage. So we wanted to see, actually, do these flies have some kind of resistance to the stress? So when we tested it, they were actually much more resistant. So that suggests there's something going on in the fly, maybe the defense mechanisms, the defense systems against these kind of bad chemicals are actually higher. We haven't done the test to see whether that's right, but these are kind of suggestive tests.
So you know they behave differently to starvation. You know that they have some kind of weird defence against these reactive oxygen species. And obviously that's in fruit flies. Is there any evidence that these processes might be similar in other organisms as well? Or is that for the future to find out?
Well, we know the calorie restriction, or what they call dietary restriction generally, has worked in everything from a single-celled yeast all the way up to mice, for sure, and even some studies going on in primates, which say that it could actually make them healthier. I saw a picture, you can see one is kind of grey and the fur is not very good and it kind of looks a bit wrinkly, and the other one who's been dietary restricted looks much healthier, and they're both the same age. So this is quite promising, and actually this whole thing feeds down into what we look at here in this lab, which is the way that insulin reacts with our body.
This is the chemical that helps us process sugar in the body, isn't it?
Exactly. So insulin is produced basically when you eat sugary foods. It helps to take the foods that you eat and store it away for later use. So you don't want to have too much sugar going around in your blood. And we found that if you have less of this insulin going around, then actually you tend to live longer and healthier. And this works in both worms, which was worked on in this lab, and also the fruit flies.
So in terms of the story that you've been working on trying to put together, what's going on in these flies with this particular mutation and why do they live longer, what's next for this particular bit of work?
Well, the next thing, so we've done all the what they call phenotype studies. Phenotype is just looking at the behaviour. So do they live longer? Are they more? Are they able to climb better? Can they move better with age? So you kind of study the differences in what you can see, and then once you've got those across, you can say, okay, I'm going to start looking at this in terms of biochemical studies. So that means testing, are they fattier than the other flies? Because we find that when we have mutants to do with insulin, we find they actually are generally fatter than normal flies.
I don't know how you tell a fly is fat.
Well, we have a test where we can test actually how much fat or lipid they have in them.
They can't just get off the ground while they're trying to fly.
No, I mean they they kind of just sit there and watch telly. You know? There are ways you can do tests to find out how different they are and find out different changes at a genetic level. What's changing? Because for example, if they have more defense, there are genes that actually protect us against the stresses that we have in our body, and you can see if the stress these genes have changed at all, see if they're higher, and that would make sense if they were.
How long would you want to live for? I mean, you're a master's student, you're a young guy. How long do you want to live for?
The obvious answer is forever, but the point is the point is you want to live as long as you enjoy the life you're in. So you wouldn't want to live in a forever if forever meant living in misery. You want to be able to enjoy the things that you normally enjoy to the extent you do now. So if I kept enjoying life as much as I do now, then the answer would be forever.
That was Michael Shanak from the UCL Institute of Healthy Ageing. Now it's time for our question of the month. Listener Anne Hawke says, when my son, who's now 23, was 15 months old, we discovered that he had neurofibromatosis type 1, or NF1, also known as von Recklinghausen disease. At the time, we were told that the next 15 to 30 years would see important developments for sufferers, but I'm not aware of any developments that could help. Please could you give me an update on the situation with this condition, which I believe is the most common genetic disease, but one that often doesn't make any headline news? Well, NF1 affects the nerves and it causes a range of problems in the body, including both benign and malignant tumours. To find out more about the current state of treatment and research, I spoke to Rosalie Ferner, Professor of Neurology at King's College London, who runs the neurofibromatosis service at Guy's and St Thomas's Hospital.
A lot has happened since I started being involved in the late 1980s. They found the gene at that time, so they know where the gene is, and they found a bit about the function of the gene. The gene product is really this tumour suppressor, so simply stated, it keeps cells controlled, so it stops them growing too much and dividing too much. The research has gone into a number of areas. It's gone into learning problems, which is one of the commonest manifestations of the condition. It's usually mild learning problems. Research has gone into tumours, both benign and malignant, and it's gone into bone. I think those are sort of the big areas. And the two things that are being done, they're looking at underlying molecular mechanisms so that they can target treatment to the particular problem. And they're also looking quite carefully at something that's not quite so buzzy but very important, is outcome measures. So it's no good having a treatment if you don't know how to measure whether it's been beneficial or not. When you look at outcome measures, you want to look at things that are objective. Have you shrunk the size of something? Have you improved intelligence? But importantly, you want the patient's point of view, because that doesn't always coincide with what you see as an improvement.
Have there been significant advances in treatment for these types of tumours and diseases?
Yes, they're starting to come through, and I think one always needs to be cautious. So if you take, for instance, the benign neurofibromas, the lumps, they cause a lot of problems because they're cosmetically difficult. They can cause neurological problems. They can cause pain and bleeding. They've used mouse models, because they're very good models for disease, to screen potential drugs that may block these rather complex tumour formation pathways. There have been a few drugs that have tried with limited success. There is a new drug called a MEK inhibitor. A MEK is just one of those signals on the pathway that activates tumour formation. Very early results suggest that this shrinks neurofibromas a little. I wouldn't say massively, but a little, so it's promising. So the hope is to increase the clinical trials for that. From the point of view of learning, they've tried statins. As you know, statins are buzzy for lots of reasons, but it was thought, again from the mouse models, that statins might help with improving learning. And they've done a couple of trials on children, and so far there haven't been huge beneficial results. What this is saying is that it's a huge step forward to understand a bit more the molecular mechanisms, understanding complications. These are early stages in treatment where people are trying drugs that are blocking different bits of this very complex pathway. There are treatments available. Are they effective? Well, at this stage, it's quite early to say.
Thanks to listener Anne Hawke and Professor Rosalie Ferner. If you've got any questions about genes, DNA and genetics, you can email me at genetics@thenakedscientist.com. And finally, it's time for our gene of the month, and it's SIRT1. One of a family of genes called sirtuins, SIRT1 is one of 7 human versions of genes found across pretty much all organisms, from bacteria and yeast upwards. Sirtuins seem to have a role in a variety of vital processes within cells, many of which are connected to energy production and ageing, as well as stress responses and the body clock. And, as you might expect, they've also been implicated in a range of diseases, including Alzheimer's, diabetes and cancer. Scientists studying ageing are particularly interested in SIRT1, as it can be switched on by chemicals such as resveratrol, found in red wine and red grape juice, and significantly extends lifespan in animals in the lab. Unfortunately, this research doesn't quite support the idea that necking a bottle of Cabernet Sauvignon every day will do you much good. That's all for now. I'll be back next month with more of the latest research into the genetics of ageing. If you've got any questions or feedback, you can email me, genetics@thenakedscientist.com. You can get in touch through the Naked Scientist Facebook page or tweet me @nakedgenetics. Every episode of the Naked Genetics podcast is on iTunes and online at nakedscientist.com/genetics. The Naked Genetics podcast is brought to you in association with the Genetics Society, online at genetics.org.uk. I'll see you next month for another peek inside your genes.